iPad and on our MSI website. So at this point, I will turn it back over to Pete, who will introduce our next speaker.
Thank you very much, Hannah. Our next speaker is Dr. Brett Baker from the University of Texas. Dr. Baker and the members, Dr. Baker and the members of his lab use omics, among other approaches, to study the ecology and evolution of marine microbial communities. Their current research focuses in particular on identifying the physiological capabilities of numerous uncultured and newly discovered branches on the tree of life. Recently, the Baker Lab has been working on studying microbes belonging to the Asgard Archaea, which are phylogenetically related to eukaryotes. Their findings have received considerable attention in the biology community as it reshapes our understanding of the tree of life. The Baker Lab is continuing to analyze the metabolic capabilities of these new archaea and doing some fantastic work in some remote regions of our planet, such as the deep sea, near and dear to my heart. Dr. Baker, welcome. The screen is yours.
Right. Thanks, Peter. Let me see here. Go. Right. Good morning, everybody, or maybe good evening, depending on where you are. Thanks a lot to the organizers and Peter for inviting me to do this. It's interesting to do this about 6 inches from where I sleep, but it's great because I think it's going to be a lot more people are going to see it. So I'm going to give kind of a broad, kind of a more big picture talk, and I will present some data, but much of what I'm presenting has been published. I am going to present some new data at risk of that being out there, but I think it'll be okay. So I'm going to start off because it's a really broad audience. I'm going to kind of give you a perspective of how I see the microbial world. So this is a really nice image, a really well commonly used image, where essentially, if you go out to the water body that's closest to you, say a lake, a river, or so on, and you stain it with DNA, and you put it under a microscope, this is what you see. So each of these little dots are microbial life. So each is an individual cell, an individual organism, species in that sample. Some of these larger dots are, phytoplankton, algae, things. I like this image because it reminds me a lot of looking up at the sky at night. And so here's an image of the sky at night. And so just for fun, I'm going to sort of use this analogy to what I do is to being, and all of us environmental microbiologists do, is very similar to what astronomers do. So just for fun, to give you a perspective, for those of you who aren't familiar, if we just do a back of the envelope calculation of the number of microbes in the oceans, the least estimate of it is that you get this really huge number that I don't even know how to say, but it gives you a sense. And if you just consider how many the current estimate of the number of stars in the universe are, you get this number. So just within the oceans alone, there's this huge amount of diversity that's out there. And really for many decades, the real basic question that us, those of us that work on marine systems and just environmental background in general, is what's out there. And so really only until recently have we been able to, we've developed tools that are able to, we've used to understand what's out there. And so what are these tools? So this, I use this analogy to astronomy. So I say that because we're very much reliant on the tools that we have as microbiologists to understand the natural world. And so for over 150 years, microbiology has relied upon cultured-based approaches, where you take your sample, you put it onto a petri dish, you put it in a tube, and you try and grow which microbes you're interested in studying. The problem is that we now know that at best, only about 0.1% of the microbes that are present in nature can have been grown in the laboratory. It's not that they can't be grown, It's just that they haven't been grown really. And so I would argue, even if you do take that organism from its natural environment, away from its natural conditions, away from the organisms that it interacts with, it's going to behave differently. So I argue that you shouldn't even really spend too much time doing this. And of course, there's a lot that can be learned by culturing something. And I'm not dissing culturing. I think it's great. And I'll show you an example of how great it is later on. But I do think that we do need to look at the natural communities to understand microbes. And so the approach that I'm going to talk about today, primarily, and it's not entirely what my lab does, but is using DNA sequencing, using genomes to understand microbial communities. So obviously, this is a very simplified version of the type of techniques that microbial ecologists use. There's several things in between here that I'm not kind of covering, but I'm just going to stick to this. So what I'm going to talk about and the methods that I'm going to discuss have really kind of come from very similar methods that have been developed in looking at the human genome. So first of all, just to walk you through this, so this is called metagenomics, which is essentially reconstructing genomes from natural communities. So first what we do is we take our environmental sample, we extract the DNA, then we sequence all that DNA, and what we get are individual small parts of that mixed community. So this just becomes a really big computational puzzle. So we use very large computers and programs to do this. And what we do is we reconstruct genomes from the original sample. So what this gives you is genomes which hadn't been known to science before. And this has been a real game changer in terms of microbial ecology, and now it's kind of spilling over into other fields as well. And this is essentially metagenomics. And what this gives us is once we have these genomes, we can use their genes, look at their genes to understand what their metabolisms are. So here's just kind of some examples from a paper a few years ago where we look and we say, okay, this organism has genes for nitrate reduction, denitrification, and has genes involved in sulfur cycling. This other organism is involved in organic carbon. We can also use these genomes to identify who's there, which I'll tell you more about in a second, and kind of their evolutionary history. Not going to get into the details here. I just want to put this up. I mean, we have these pipelines within my lab where we go through and we sort of do all this. The other thing I want to touch upon is we also, we don't just rely on genomes, of course. Genomes are really just kind of the foundation, kind of a framework for understanding things. We use other approaches like DNA SIP and BondCat, and collaborate a lot with Roland Hodson-Pickler to do these approaches in the deep sea at the moment. So kind of to give you a perspective how this has changed things. So if we look at the types of DNA sequencing approaches that have been around and how it's changed. So essentially, when genomics was first being done around in the early 90s, we could only get about one genome at a time. And then first metagenomics was first being used. We were able to get dozens of genomes. Jill Banfield's lab at acid mine drainage. I'm kind of the first to do this. Now we're at the point where we're reconstructing thousands of genomes from an individual sample. Okay, so this is absolutely mind-blowing to me because when I was in grad school, it just we couldn't even imagine that we'd be able to do this. And so kind of a project led by Jill Banfield's lab and Laura Hugg's first author kind of asked the question, what are all these new genomes doing to the tree of life? And this is what I like to refer to as the Hubble moment, where the Hubble telescope took these images of deep space and saw things that we hadn't seen before. This metagenomics approach has really changed the tree of life in that very similar way. And so here's sort of an updated tree of life. And so for those of you who aren't familiar with looking at these, essentially each one of these branches, each one of these names is a phylum, and each one of these branches is the length of the branch is the distance in the DNA sequence between any of those two organisms. So you see eukaryotes down here, you'll see archaea here, which are closely related to eukaryotes, which I'll talk about later. And each one of these names is a phylum of life, okay, as distant as any taxonomic group can be. And anywhere there's a red dot is a phylum that has not been cultured, okay? So this has completely expanded our view of the tree of life. And I like to use this quote by a colleague at UC Davis and Jonathan Eisen, when this paper came out, there was a press release where he said, "This is humbling because holy shit, we don't know virtually nothing about the biology of most of the tree of life." And that's true. We now we have this new view of the tree of life, but we don't really have a very good understanding of the biology yet. And so just to kind of reiterate this, so if we look at those groups that haven't been cultured in a laboratory, haven't been grown in a laboratory, it shows this here. And so even within those groups that have been cultured, there's huge numbers of organisms that, so even the ones that aren't black here that have culture representative groups, there's huge numbers of taxa that haven't been cultured as well. So there's this huge limited understanding in the tree of life. So my lab primarily works on marine sediments. And so we study marine sediments because it's really important in carbon cycling on the planet. And that's because atmospheric CO2 is taken up in the surface oceans and eventually settles to the surface floor and the ocean floor. And as a result, the marine sediments contain the largest pool of organic carbon, right? So understanding the microbes that live in these marine sediments is really important in understanding how this carbon is processed there. And so if we just look at an updated tree from this year of the Archaea, here shown, and again, those in red dots are those that have been cultured. And if we look at the dominant groups within marine sediments, they primarily have not been cultured. So our understanding of this really important, huge part of the planet's biosphere, we don't have cultures for, we don't really understand yet. So just to briefly walk you through some of the sediment projects at my lab, we work off the coast of Oregon. We do a lot of work in estuaries. We've done a lot of work in the Gulf of Mexico, close to where my lab is. Again, in estuaries close to my lab. We work on estuaries in the San Francisco Bay with Chris Francis. And primarily what I'm going to talk about today is the Guaymas Basin, where it's in the Gulf of California. It's about 2,000 meters where there's these hydrothermal vents. And so if you go down to the, once you go down in the submarine and you get to the surface, the ocean floor at Guaymas, you see these really colorful mats. And essentially this is just microbes that are thriving on these sediments because there's all this really interesting chemistry that's going on there. I also show this because it kind of highlights how dynamic this environment is. I mean, a sample here collected here is very different than a sample collected here. So we've been kind of really putting a lot of time and effort into sequencing different sites at Guaymas. And so we now have around 31 different communities that we've sampled. The other thing to keep in mind here is that these sediments get, in certain places, not everywhere, they get really hot as you go down. So you can just go down a few 30 centimeters, you can get up to 100 degrees centigrade. So that's a big part of the influence here of these communities. So people often ask me, have we found all the new phyla? Do you stop finding phyla? And so here's kind of a new tree based on a recent sequencing effort in my lab by Shenzhou and JD, who's a really talented undergrad in my lab, and Catherine, who's a grad student, are working on this. And if we sort of look at the new diversity within our new sampling approach, we find that there's a ton of new phyla. So it's kind of astonishing because the more sequencing we do here at Guaymas, the more diversity we find. It doesn't seem to really end at this point. So from this recent effort, we've got around over 13 new bacterial phyla and 6 new archaeal phyla that we're working on. And so one of these new archaeal phyla that we're kind of working up at the moment is what we've named Brock Archaea, which is named after Thomas Brock. He's a very famous microbiologist who worked a lot on hot springs. And so here's a tree just showing where they lie within the Archaea. So if you're familiar, this is the Thalm Archaeota. They fall within the Thalm Archaeota, and they're here. They have very unique physiology. So if we look compared to, this outer ring is comparing the gene content of these Brock Archaeota to related Archaea, and then we find that they're quite unique. We also find that they're really widely distributed. and especially within hot springs. And so that's kind of why we got the name Brachioarchaea, because Thomas Brock did a lot of work at Yellowstone hot springs. And so here's just a map showing of where we've gotten different genomes that belong to Brachioarchaea. So what are Brachioarchaea doing? They are pretty interesting, actually. So they're primarily involved in C1 we think they're involved in using C1 compounds like methanol and methylamines, as you can see here. It's not very clear what they're doing, how much energy they're getting from these compounds, or how much they're incorporating into biomass. But what is unique about them is that if you consider most of the, so Brachioarchaea are unique because most of the organisms that use these C1 compounds convert it to methane. And Brachioarchaea doesn't do that. It converts it into different things. So essentially, methylamines are very abundant in aquatic systems. So in marine systems, in hot springs. And people have shown, primarily Mandy Joy's lab, that these methylamines are used quite a bit by geochemical techniques, but they don't usually convert them to there's a huge portion of that don't get converted to methane. So we think that Brachioarchaea plays a really important role in carbon cycling. So kind of taking a step back, that's kind of like looking at a new phyla. Now if we just look at like a phyla that's been cultured, and for instance, the Delta Proteobacteria, this is work by Maggie Langweed in my lab. So this is essentially, we've taken all the new genomes from within the Delta Proteobacteria and add them to the Delta tree, and we have all these new branches within the Delta Proteobacteria. So this is essentially resulted in roughly 28 increase in the genome diversity just within Delta Proteobacteria. And if you look at what these genomes have, they're quite a bit different from those that are cultured. So here on the left are kind of the cultured Delta Proteobacteria, and here on the right are uncultured types. And you can kind of tell that there's a lot of different things that are showing up in terms of metabolism compared to those that are known. Let's skip that for time. So what does this all mean before I kind of switch gears and talk about Asgard Archaea? The point of this is we're trying to get an understanding. There's this black box in terms of our understanding of the microbes and sediments. And so we know what goes in. We know what organic compounds that go in. We know they get processed, but we don't really know who's doing it. So what we're trying to do is reconstruct all these genomes and understand who's doing these really important processes. And so this is kind of an overview of just Archaea, our understanding of the metabolisms there. In the end, what we'd like to do is understand how all these organisms interact and how carbon and energy kind of flows through marine sediments. And so this is an overview just showing of Archaea's role in sediments. And you can see that, a lot of these uncultured groups have really important roles that really weren't appreciated before. Okay, I'm going to switch gears and talk about Asgard, as Peter mentioned. I think this is probably one of the most exciting things to come out of metagenomics in recent years, and it's really changing our understanding of a lot of things. So this paper, all this work that I talk about here is a collaboration with Thijs Etma. So his lab in 2015 found these organisms near Loki's castle hydrothermal vent in North Atlantic, and he named them Loki Archaea after Loki's castle. Around that same time, my lab had gotten some genomes from an estuary, and this paper came out. I went running in my grad student's office, Kylie Seitz, and we realized that we had something really interesting. And not, we knew there was something interesting, but it was interesting for reasons that we hadn't appreciated before. We weren't really looking for the origin of eukaryotes, essentially. So what's cool about these organisms is if you put them in the tree of life, here's Archaea, kind of showing the same tree I showed now, but in a linear fashion. The eukaryotes fall, they kind of get sucked into the Archaea, and that's because they are related to eukaryotes. So us, trees, and other things are related to these organisms that we call Loki and Thor. So Thijs's lab and myself and some others kind of set out to assemble more genomes from different environments. And this kind of led to more these what we call Asgards, after Norse gods being discovered. So we have the original Thor and Loki that I mentioned before. We have this new organism that we call Odin Archaea, and another organism that another phylum called Heimdall Archaea. So this is a huge effort to get to make this very robust tree that Tice at my lab has put together here. And what it tells us is that Heimdall Archaea appear to be the closest to eukaryotes. This is very important because it means a lot in terms of our understanding of your early eukaryotic evolution, because it means that eukaryotes shared a common ancestor with Heimdall at some point in the past. So my main message today is that we are Asgardian. And some of you may ask, why haven't we seen some of these before? So like the Thor, for instance, hadn't been picked up in diversity surveys. And those of you familiar with PCR diversity surveys, you use primers. And here's a look. Essentially, these organisms have very different, they're very unique 16F sequences, so they weren't picked up by these PCR primers. The Thor has 3 mismatches to really common primer. So it's pretty exciting that there's something that's related to eukaryotes that's been found. And if you look at the genomes of these organisms, they have a lot of proteins that have only been seen before in eukaryotes. And so here's just kind of showing an overview of these. So at the top here are these Asgard Archaea, then eukaryotes, and then the distribution in Archaea. So you can see that some of these proteins that are involved in information processing, cell division, trafficking, and so on, have been seen in Archaea before, but not to the extent that they're present in Asgards. All right, so this kind of reinforces the idea that these Archaea shared a common ancestor with eukaryotes and us. And so if we step back and say, okay, well, what does this mean for our understanding of the origin of eukaryotes, essentially? So at some point in the past, we have this Archaean, which is related to Asgards, that merged with a bacterium and formed the first eukaryotic cells, okay? Again, I want to make this clear, you know, these Asgards are not the archaeal host in this relationship, but they're descendants of that, right? And so in understanding how eukaryotes came to be, we want to understand what the relationship was between this Archaean, this Asgard, and a bacterium. Why did they have this relationship, and why did they merge to become the first eukaryotic cell? So it has huge implications in understanding the origin of eukaryotes, essentially. It's a symbiotic relationship. So to understand this, a study led by Tice and Anya in Tice's lab, we looked at the metabolisms of these different Asgards to see what their metabolisms are and what this interaction might be. So based on this, you know, the genomes of these organisms, we came up with this new model of essentially the symbiosis of the origin of eukaryotes, essentially. And we hypothesized that it was, the interaction was essentially held together by a transfer of hydrogen or formate or acetate, or it could be electrons, essentially. These Asgards, and particularly the Heimdalls, really are kind of heterotrophic. They can only eat organic carbon compounds. So we think they're amino acids and other organic aromatic compounds. And then very recently, very excitingly, it was published this year, but it was announced last year that someone actually cultured one of these Archaea. And so prior to this, we only had genomes. We didn't know what they looked like really. And some people had in the field had even guessed that these were just some sort of artificial construct that was being created. So it was nice that someone actually was able to grow one of these in the laboratory. This is a huge effort by a large group of great scientists in Japan. So here, essentially what they did was this really confirmed our hypothesis that this interaction between an Asgard, or the Archaean, and the bacterium was based on hydrogen and formate. The images of these are really exciting to see. They form these really large appendages, which are hypothesized to be part of this interaction as well. This is fantastic. And I just want to remind everybody that this Loki isolate is one of many Asgards that are out there. All right. And so here's an updated genome tree of Asgards that we have in my laboratory. And so you'll see here on the right, you can see Loki Archaea. It's unfortunately I don't have the isolate in here, but it's it's deep within this tree. And remember, the phylogeny is telling us that eukaryotes branch somewhere within Heimdall. Okay, so if you're trying to extrapolate metabolism within this organism, which is a totally different phylum over here, to understanding the origin of eukaryotes, it's a bit dicey. However, it does show and indicate that this symbiotic relationship is probably widespread within the Asgards. And so as you can see, we have a lot of new groups, and some of these are new phyla, which we're working on. I won't show too much today, but there's some really exciting things going on in terms of phylogeny, metabolism, that's advancing things. One of the things that I will talk about is this new group, this new phylum that were named after Hela, the goddess of the underworld. So its name is Hela Archaea. Here's Hela in a tree with the Asgards, and it's related to Loki. And if you look at its eukaryotic proteins, it has a very similar composition of eukaryotic proteins as Loki itself. So again, another organism that appears to be involved in some sort of symbiotic interaction. So this is Hela's metabolism, and based on its metabolism, we believe that there's a hydrogen-based interaction. It's pretty clear to us that they're using butane, they're doing anaerobic butane oxidation as a syntrophic interaction with an unknown partner at this point. Okay, so I want to finish up there. These are the people in my laboratory. Jill Banfield and my Chinese collaborators are involved in the Brock Archaea paper and the Tree of Life, obviously. Thijs Etma, probably one of the best collaborators I've ever worked with, and everybody in his lab, Anya, Laura, Daniel, Eva, and so on, were a big part of this work. Andreas Teskey is kind of spearheaded all the Guaymas work and has been a good collaborator on many of these projects. And I'm funded by NSF and more recently the Simons Foundation. So thanks.
All right, thank you very much, Brett. And congratulations, Brett, on the recent Simons Foundation Award. Nicely done.
Thanks.
All right, we have some questions for you. Let me check things out over here. Okay, so start with a very big question. In general, why can't we culture so many of these species in the lab? Is it nutritional or extreme temperature requirements? So that's the question. And perhaps while you're at it, you can even talk about species.
Yeah. So in terms, I mean, well, I think it's nutritional in some environments. In terms of marine sediments, I think the main limitation is that things grow really, really slow, especially in deep sea sediments. So for instance, as I mentioned, the Lokis, their doubling times is, I can't remember what it is offhand, but it's weeks. So you're talking about, to culture that particular organism, it took them 12 years. So I think that's the main hurdle. Yeah. Is there another part to that question?
Yeah, no, that's fantastic. So tell us, what are your thoughts on this idea of species among microbes? What are some of the challenges with the species concept?
Oh boy. I get antsy when people ask about species because there isn't really a good answer. And
there isn't.
I think we're starting to see within genomes, we are seeing that there is a clear delineation at the species level. What that delineation is, no one is agreeing on at the moment, but I think hopefully it will become more clear. Yeah, species is a continuum. That's what Dave Stahl always used to tell me, and I think that's probably true. Yeah, I don't know.
Indeed. And I'll ask the 1,000+ people who've registered here not to quote Brett on this.
Oh, yeah.
Don't put him in the hot seat. We have some more questions, Brett. What clues from Brock Archaeota's genome help explain its thermophilic lifestyle?
You know, that is something, man, I wish Valerie could answer that question. I think we had discussed it early on, and we hadn't, honestly, I don't have a good answer to that right now, so I'm not going to start BSing. Yeah, I don't know, to be honest.
Yeah, fair enough. Work to be done, right?
Yeah.
Can you speculate on what the first interaction between the archaea and bacteria may have looked like? Do you think there was a predatory or a mutualistic aspect? Just speculating.
Yeah, I mean, so, again, we don't really know, but it's from what we can tell, the predatory hypothesis really hasn't come to fruition in terms of gene content. And as I mentioned, we know very clearly now that Loki is a syntroph, and it appears that other organisms are very likely Heimdall, very likely Hela,